Hello, my name is Neil Boudville and I am Professor of Renal Medicine at the University of Western Australia Medical School. And today I'm going to be discussing the different ways that we can measure kidney function. And I'm specifically going to cover urea, creatinine and estimated glomerular filtration rate. So in our time together today, I'll be describing the metabolism of urea, the metabolism of creatinine, and understand how your estimated glomerular filtration rate is generated by the laboratories. And with all of this information, I'm hoping that we'll be much better at interpreting these markers of kidney function. So let's start with urea. Urea is this molecule here, and it is created through the breakdown of amino acids primarily occurring in the liver, which generates ammonia that then enters the urea cycle to form urea. Blood urea nitrogen is another term that you may have heard of that's commonly used in the United States and North America. And this is also another measure of the amount of urea in the bloodstream, but it is measuring the nitrogen components of this molecule as compared to the urea concentration, which is the entire molecule. Urea that is generated by the liver is then excreted from the body by the kidneys. So this is the ammonia that is generated by the degradation of amino acids in your liver, which then enter your urea cycle, leading to generation of urea molecules that enter the bloodstream and are subsequently excreted from your body by your kidneys in your urine. Urea is freely filtered in the glomerulus. However, it is also reabsorbed in the renal tubules, especially in the collecting ducts. The amount of reabsorption is often dependent upon the amount of time the urine spends within the tubules. So the longer the amount of time, that is, the lower the flow of urine or the lower the amount of urine is made, the greater the amount of reabsorption will occur, leading to an increase in your serum urea concentrations. Let's move on to creatinine. Creatinine is created by creatine. So to understand creatinine, we first have to go backwards a step and understand the synthesis of creatine. Creatine is synthesized from two amino acids, arginine and glycine, in the kidney, and then in the liver, via these enzymatic pathways, you generate the creatine. You don't really need to remember this pathway, you just need to understand the origins of these various things. Creatine formed in the liver will then be acted upon by an enzyme called creatine kinase, which creates creatine phosphate. Creatine phosphate is the store of energy in skeletal muscle and the brain, allowing the rapid generation of energy in ATP. Both creatine and creatine phosphate is degraded to creatinine. And it's this creatinine that then passively moves into the blood, and this creatinine circulates in the blood is approximately proportional to the amount of skeletal muscle that a person has. Creatinine in the blood is freely filtered in the glomerulus of the kidney like urea. It does not undergo any metabolism within the human body. And in this diagram here, you can also see that there is a little bit of secretion of creatinine into the kidneys via the tubules. There's actually, even though these arrows seem pretty big, there's actually only a very small amount of secretion of creatinine in the kidneys compared to how much is filtered. So in most cases, it can be ignored, and we could just think about it being filtered. Where it becomes relatively important might be in situations where there's very little filtration occurring, as in someone with severe kidney failure, so that the amount of secretion relative to the amount of filtration is a bit closer to one another. So you can imagine where there is reduced glomerular filtration, there will be reduced creatinine excretion, and creatinine will build up in the bloodstream. The higher the creatinine, the less the glomerular filtration, or the less the kidney function. Now let's finally talk about estimated glomerular filtration rate, or eGFR. This is a number that's generated using a mathematical formula that just includes the patient's age, their gender, and their serum creatinine. So just these three things. If they're female, this is the equation that is currently used in Australia, and if you're male, this is the equation that's currently used. It has to be remembered that this is only an estimate of the kidney function. We mustn't believe too strongly in the absolute number that is generated, but it does approximate kidney function. It is, however, useful as a baseline for patients, so you can use it to compare changes in their kidney function over time. So how do these three measures of kidney function compare? Well, urea is not a good marker of kidney function. It can be elevated for many other reasons rather than just reduced glomerular filtration. It can be increased because of increased production of urea, which occurs in the context of a gastrointestinal bleed, when you're using steroids or in other catabolic states, or if you have a particularly high protein diet. It can also be increased due to the increased renal reabsorption of urea, which occurs with dehydration and heart failure. Creatinine is the preferred marker of kidney function over urea, but it also has some limitations. And the limitations are around the fact that the relationship between serum creatinine and kidney function is not linear. So in this figure here, you can see on the x-axis is the serum creatinine, and on the y-axis is your kidney function or glomerular filtration rate. So let's look at the blue arrow first. And this depicts a person who's got a relatively low serum creatinine or relatively good kidney function. And in this situation, for a set increase in serum creatinine at this low baseline or good kidney function baseline, you get a large reduction in kidney function or large reduction in glomerular filtration rate. For the same increase in serum creatinine, but where the baseline creatinine is much higher or the kidney function is poorer, this same increase in serum creatinine equates to a much smaller reduction in kidney function or glomerular filtration rate. So the relationship between serum creatinine and GFR is not linear. And this is why your eGFR is easier to understand. First of all, an eGFR of 30 mils per minute per 1.73 meters squared can be translated to meaning that there is 30% kidney function, as a normal GFR is between 90 and 120 mils per minute. In addition, halving of your eGFR means that your kidney function has gone down by a half. So it's a lot more understandable for health practitioners and also for the patients. So hopefully I've demonstrated to you how urea and creatinine is metabolized or dealt with within the human body, and how the estimated glomerular filtration rate is generated. And by understanding these processes, I'm hoping that you understand the usefulness and weaknesses of these three measures and how your eGFR can really help in discussing someone's kidney function with the person. Thank you.